Hi, my name is Hannah Mahoney, and I'm going to be discussing the impact of female choice on egg viability in the American dog tick, the Dermacentor variabilis. Myself, Kenan Oyen, Melissa Kelly, and Joshua Benoit all worked on this project through the Department of Biological Sciences at the University of Cincinnati, Ohio. First, I'm going to be discussing the effects of relative humidity on tick biology. During short dry periods, the water uptake following dehydration reduces energy reserves. Multiple dehydration bouts alter starvation and longevity. Dehydration primes ticks for water vapor uptake. Short dehydration bouts increase tick activity and decreased exposure to fungal pathogens. During humid periods, hydration can occur immediately following questing. Starvation is extended with increased potential contacts with the host. There are increased survival of juvenile stages, along with increased probability of egg deposition in human environments, and increased exposure to fungal pathogens. During drought, there's reduced questing, lower survival, increased nighttime activity, reduced host contact, decreased soil moisture events water vapor uptake, along with decreased exposure to fungal pathogens. The introduction to our experiment, the Haller's organs are located on the front legs of the tick and are involved in chemical sensing. Interference with this organ alters tick behavior, and in this study, we examined the Haller's organ for the ability to detect humidity. The figure just shows the Haller's organ of the tick. Like I said before, it is located on the front legs of the tick. Since the Haller's organ is associated with chemical sensing, we studied if the Haller's organ can be associated with humidity detection. Humidity sensing impacts biology, the tick biology in multiple ways, such as their hydration and energy conservation. The Haller's organ could be associated with these biological processes, such as tick reproduction. If the Haller's organ is associated with humidity detection, then that gives us insight to egg viability in different relative humidities. So just a background on what we're studying, the American dog tick, which is the scientific name is Dermacentor variabilis. D. variabilis can be located in regions of eastern North America, and it can act as a vector for diseases such as the Rocky Mountain spotted fever. This next slide explains the alteration to Haller's organs and the humidity attraction. So the first figure shown is a setup to determine tick attraction to relative humidities. These tubes were 40 centimeters long, and each of them correlates to what alteration to the Haller's organ there was. 0 centimeters is closer to 0% humidity, while 40 centimeters is closer to 93% humidity. So these tubes, like I said before, correlate with the different alterations. So if we burned off the organ, if there was wax on the organ, if the organ was cut off, there was also a control group, and then there was also a group of ticks that were control for 2 days, and then they put wax on them. So in this table, you can actually see the control to wax. Actually, they decrease in humidity where they lay their eggs after they get wax put on them, even though they were a control subject to begin with. When the wax is removed, you can see that the humidity detection goes back up to almost normal like the control. When the organ was burned, cut off, or wax was placed on it, the humidities dropped to where they laid their first egg. So the message for this slide is just the Haller's organ is responsible for chemical sensing and is very sensitive to alteration. This is important because it can be responsible for female ticks depositing eggs at desirable humidities. Materials and methods. So these three petri dishes just show the different groups of 1, 5, and 50 eggs. So each of these were examined, and examining just included looking for larval emergence or if the eggs were dead. So these 3 petri dishes were held at different relative humidities, ranging from 33 to 100%, and the viability of eggs was assessed by the examination of larval emergence. Egg viability increases with larger treatment groups. The tables show 2 sample groups, n equals 1 and n equals 50, at a relative humidity of 100%. These figures depict that egg viability increases when the treatment group has a larger number of tick eggs. At n equals 50, there is a higher average of larval emergence than at n equals 1. So essentially, egg viability positively correlates with the number of eggs in the treatment group. The average number of larvae increases when the treatment group has a larger number of ticks. And this is actually due to ticks preferring to lay their eggs in bigger clusters because it proves for a higher viability of the eggs to become larvae. The next thing I'm going to discuss is the egg viability at different relative humidities. So as you can see, we have humidities at 33% relative humidity, the egg viability at 75% relative humidity, and the egg viability at 93% relative humidity. So the average of the egg viability in groups of 50 eggs at 33%, 75%, and 93% relative humidity. Survival is increased with a higher relative humidity. A t-test was ran for each set of data, which proved significance in the averages of the larvae and dead eggs, and all comparisons proved to be significant. Egg viability does positively correlate with increasing humidity. So as you can see in the first table of 33%, there were actually no larvae that emerged at this relative humidity while all of the eggs were actually dead. So at 75%, you can kind of see that there is some larva emergence, but not as much as 93%, and it's mainly just dead eggs. And at 93%, you could see that the average is much higher of larval emergence compared to the dead eggs found at this humidity. So in conclusion, the Haller's organ is likely the site of humidity detection in ticks. Humidity detection allows females to deposit their eggs under favorable conditions. The results of our study can help improve the understanding of Dermacentor variabilis, their preferred microhabitats, the trends in reproduction, and new methods of control. Acknowledgements, I would just like to acknowledge Melissa, Kenan, and Joshua for their contributions and edits made to this project. And I would also like to acknowledge the undergraduate students who played a role in this experiment and made it possible for me to complete this project. Thank you very much.